to you all who are joining us. Today's webinar is about 7 pitfalls to avoid before you purchase a single-use flowpath chromatography system. And a lot of these are being done today for gene therapy or people who are doing mRNA processes. So again, thank you for joining us. We'll get started now. And so just a little bit about Sepragen. We are a company with a 30-year history. So it's a company, we are a company that has been around for a while. Our products are used in the manufacture of 30 or more FDA-approved drugs with over 200 global installations, and including 2 major COVID-19 vaccines that are produced using our automated systems. So we know something about GMP and large-scale manufacturing as well as small-scale manufacturing. And in fact, during the COVID crisis, we helped a company go from 10+ million doses a month to over 100 million doses a month, and making integrated chromatography systems with buffer blending capability. And our products today, equipment is used in the manufacture of biological drugs and vaccines exceeding $20 billion of material that flows through our equipment. And we are inventors of radial flow chromatography columns. If some of you have not heard of that, definitely check us out. They can be used with any resins and enable about 4x the throughput, 4 times the throughput of conventional columns, a whole line of chromatography systems and skids. And then today's topic about single-use chromatography systems. And then if some of you are contemplating continuous chromatography with 2 or 3 column SMB type systems, we have those, and also those in single use and buffer dilution and inline buffer blending systems. And then integrated systems. We are the first ones to do this, where you've integrated like buffer blending into chrome systems or into continuous chrome, or integrated multiple chromatography steps like a flow-through step along with capture steps in chromatography. Okay, and these are some of the customers who've used our products over the years. You'll see some well-known names. And now we'll get on to the main topic of our conversation, which is single-use chromatography systems. And so firstly, why do people use single-use chromatography systems? Well, for one, there's no need for cleaning validation, right? You take out the flow path, almost like the tubing around a patient at the hospital. You remove all the IV and everything else, and you toss out the tubing, whereas the pumps and the monitors and all that stay by the patient bedside. And that way you can have multiple products that can be run in the same facility, multiple batches, and there's no chance of cross-contamination. And of course, because the flow paths come sterile, pre-sterilized, no bioburden issues, very suitable for cGMP production. You can qualify it for multi-product facilities, batch to batch. There's no cross-contamination. And then these, simply by changing different size flow paths in a system, so you might, in a bench scale system, you might have a 1.6 millimeter diameter flow path or a 3.2 millimeter. And depending on the pump ranges, you can go down as low as 0 to 5, 10 mils per minute. And of course, as high as we have systems as large as 60 liters a minute, right? So the whole gamut.
Hey, Vinay.
And some of these
Vinay,
have
I'm
been...
sorry to interrupt, but the participants can't see the slides. So would you be able to put in presentation mode?
Oh, I thought I did. So can you see my slides now?
Yeah, now we can.
You can. So let me see.
Do you want
I
to just go on?
apologize.
Yeah.
I
You want to just go on
apologize for that.
slideshow? It'll show the entire slide then.
Yeah.
Yeah.
Can you see now? Hello, Sanjit.
Yeah, this is better.
Okay,
Yeah.
fabulous. So sorry about that, guys. Some technical glitch. Anyways, and so with some of these systems, you can also have single-use systems which give you the ability to do inline dilution. So you can, make one batch for of buffers for a campaign, concentrated buffers, and then connect Wi-Fi and dilute everything online. Or you can even make buffers from stock concentrate like monobasic, dibasic, phosphate, salt, and water, and do that in a single-use system as well. So anyways, before I I'm jumping too far ahead. These are some of our products, the radial flow columns, going again from the bench to very large scale manufacturing, multi-use chromatography systems like this QuantaSep system you see here in the center, and to the right you see single-use systems. And we have again multiple single-use systems with different flow paths going from bench all the way to large scales. We have ion exchange resins. We also customize chromatography systems. So unlike other companies that have like 3 or 4 basic models and you fit your process to those specific models, we are not a one size fits all company. And so we customize to your size, your specification. Somebody may want pre-column sensors. They may want pre-column filters. They may want to communicate with multiple with tanks in a tank farm or in a different room, or and look at levels of different tanks, so we can work with you on all that. And on the right here you see a single-use continuous chromatography system with 3-column with a 3-column SMB system where you're loading one column flowing onto the second column while the third column is going through the wash and elute. And this has integrated buffer blending, but it also has a single-use flow path. Okay? And then some very large systems. And then here, of course, you see on the left our fusion single-use system, which is a continuous bench scale system, which runs between 0 and 100 mils a minute or 0 and 300 mils a minute. We got large continuous systems. We got buffer blending systems, automated column packing systems, and the like. But focus strictly along single use. And I'll get to what the 7 pitfalls are. Those are very, very important as you go to looking to purchase or evaluate a single-use chromatography system. So we've got a 20-liter per hour system, 100-liter per hour system, 600-liter per hour systems, 1,000 liters per hour, 3,600 liters per hour. And all of these are all single-use flow path systems with disposable sensors, pump heads, flow paths come sterile, etc. Right? And so they have a very smart GMP 21 CFR Part 11 compliant software. The materials used are USB grade 6 ADI-free materials, and the flow paths are fully integrated and can be installed in about 15 minutes. And you've got your standard sensors of pH, conductivity, UV, flow, pressure, air, etc. Right? And of course, then you have these continuous single-use flow path systems, as you can see here in the in the software screen at the bottom. Where in the orange you have the load, and then you've got the 4 pumps in blue at the bottom, making the buffers and delivering to one of the 3 columns. We can discuss all these in more detail, but I really want to get to what our topic is today, which
Vinith,
is yes.
It's still not showing. It's still frozen. Would you be able to go in slideshow mode? Just click on
Wow.
slideshow at the top.
I am in the slideshow mode. Which screen am I frozen on?
Right now, I'm seeing the seven. What are the seven common pitfalls? Slide.
Okay, so I can just use that slide. I can go to slideshow mode, but it seems like I'm not transitioning to the next one. Can you give me some feedback and see if I am, if you can see the
Mm,
whole slideshow now?
Yeah. Now I'm just seeing a black screen, and it looks like you've gone off slideshow again. You're back to home.
Wow. I don't know what the technical glitch is, guys. So maybe I'll just work from this screen for right now and not take up people's time. Um, I apologize for this technical glitch. In any event, thrown me off my stride. But here are the 7 common pitfalls that people experience or should really look at when they're considering chromatography, a single-use chromatography system, right? One is, and especially for viral vectors and mRNA, and I'll explain to you why, because a lot of chromatography, single-use chromatography systems have been developed for mAb or large-scale usage, right? And particularly on the small scale, the sensors, the accuracy, the disposability, and the design is not really suited for small scale, which is what viral vector production and mRNA production is all about, right? So a lot of times the question is, do I really need a single-use chromatography system for lab purifications? All the systems that are out there, single-use systems, are too large or have large holder volumes. Are there chromatography systems suitable for viral vectors or mRNA production? Then things like, how do you deal with air bubbles in a chromatography system, right? In larger systems, you may have bubble traps. Well, large holdup volume. So we'll discuss that. And how easy or difficult is it to change flow paths? And then scalability. What are the minimum and maximum column sizes that are covered by the vendor single-use chromatography systems? Then if you're using single-use chromatography systems, how do I know if there are leaks? What do I do with that? If I'm running an overnight run, can I lose my batch? And then will these systems enable me to do chromatography runs with inline buffer dilution? So I don't have to make buffers each time. I could make stock solutions or concentrates and just dilute them online and use them like any other, and makes it easier. So instead of making buffers for each run, you could make buffers potentially for a campaign, right? But make it 10x concentrated. So we'll address each of these pitfalls one by one. And the first one is, do I really need a single-use chromatography system for lab scale purification? Well, the answer, of course, is if you're making GMP production, GMP material, the answer is yes. Because otherwise you're compromising, you're using non-GMP lab scale systems to make GMP material, and they're not necessarily sanitary. And then can you scale up protocols? And then, of course, if you use a sanitary cGMP lab scale system, even though it's lab scale, it's being used for production, you can scale up to larger scales, right? You don't have to worry about endotoxins or bioburden. Whereas with standard lab systems, you have to worry about cleaning, you have to worry about endotoxins and bioburden. And then, of course, you're going to be using them multiple sometimes. So you have to do cleaning validations. Whereas here, If it's multiple products, you can segregate flow paths for different products. Even if you want to reuse it, you can still clean it out with sodium hydroxide and reuse it. But one, for instance, flow path might be for lenti and another one for maybe for AAV, right? So you could use both, but then you've got separate flow paths that you're using for separate situations. And the other advantage of smaller single-use systems is that even if you have large-scale systems in manufacturing, then you can do scale-down studies and troubleshooting with smaller single-use systems, like what we show here is an Adept 300 SU. And that's a system with two flow paths, 1.6 millimeter, which can go between zero, which allows you to run between 0 to 100 mils a minute, single use, right? With all disposable sensors in the lot. And then in the same machine, you can put a larger flow path, which is, 30 ml to 300 ml per minute, right? So again, very, very low hold-up volumes, completely sterile, sanitary, that you can use on a very small scale, and perfect for, CGMP manufacturing. If you've got a 20-liter bag, 50 liters, or, even smaller bioreactors, or in the case of mRNA, the small IVT reactor, okay? And then, of course, you have full documentation, right, which will be part of your CMC. So you've got full documentation, which is GMP grade. And then the methods can directly be translated to larger and larger systems, right? Because they're all based on the same software platform. And then pitfall number 2, are these single-use chromatography systems and continuous single-use chromatography suitable for viral vectors or mRNA production? Well, as I said, the flow rates are very small, so they are suitable. And some of the other competitors, I think one of the things one should really look at is, are they really suitable? Because they may have smaller systems, but then the flow rate accuracy that they provide on these smaller, meaning lower flow systems, is simply not there, right? For instance, the pH accuracy may not be there. We have one vendor that says that their pH sensors are on a small scale of plus minus 0.3 pH. Well, that's a huge pH variation, right? Or the accuracy of plus minus 5% conductivity, or a flow rate accuracy of plus minus 12 ml per minute. Well, if you're running 50 ml per minute, plus minus 12 ml per minute is, huge variance, right, in the flow. So those are important things. Just because somebody has a system doesn't necessarily mean it's suitable. Whereas you can see the accuracy that we provide in our small scale systems mimic the accuracies that are there in the larger scale systems. So you can have very high levels of performance on a small scale as well. And then pitfall number 3, what about air? Right? So some people might, if air bubbles get in, they may just kind of stop the process and give an air alarm. Somebody else might put a bubble trap in there. But if you have a bubble trap in the flow path, well, guess what? You just introduced a huge holdup volume. And, I don't know if you can see in this little chart that we put up in this chromatogram, on the left is a peak without without any bubble trap, and on the right is what you get if you have material going through a bubble trap, right? Because you have a mixing of two fronts between different buffer changes. They're not sharp. Cleaning validation is an issue if you wanted to reuse, loss of materials, and certainly large holdup volumes. So Sepragen systems have unique auto air eject systems, so we can measure the size of the air bubble and dynamically eject it from the system, right? So no air enters the column, but you're doing it without introducing hold-up volume in the system, right? Which is causing unnecessary gradient formation or product dilution, right? We can sense the air bubble and eject it out of the system in real time. So it's a more elegant solution of dealing with these kinds of issues. So checking out how somebody's system deals with air bubbles is very important. And then what about leaks? Sometimes you have systems doing runs overnight, right? Or unattended while the operator is doing other things. And with single-use systems, you've got lots of connections. You're running through a peristaltic pump, very often small peristaltic pumps. The tubing might wear out or get pinched. And so, you could have, if it gets pinched, you could have high pressure. You certainly can have high pressure alarms, and that'll trigger the system to stop. But what if there's a leak in the tubing or in the fitting? And usually you find that out too late because there's a big pool in front of your machine, or there's a loss of a batch. So all our systems, so the solution is that all our systems have a low point. They have a catchment below the tubing set. And so if there's more than 3 to 5 ml of liquid that leaks, the system will give you an alarm. There's a leak sensor. The system gives an alarm and will pause for the operator to fix the problem, clear the condition, and then resume operations. So that way you avoid any catastrophic batch loss. And the next pitfall that one should always consider is scalability. You may have a large-scale single-use system, or you may have a right size for a particular process, but what happens if the process is altered or changed? What if you decide to use it in a multi-product facility on different other products? Then does your vendor have the range of machines that could provide you with columns where you can use columns of different sizes? And so the first thing is, in the same machine, can you use columns of different sizes? And then can you go all the way to most commonly used sizes from across different systems? So we've shown you here with Sepragen's QuantaSep Adept single-use systems that you can run columns as small as 1 centimeter diameter columns, right? All the way to 1 meter columns with a line of machines, right? So there's the 300 SU, which can run between 1 centimeter and 10 centimeter diameter columns, and the 3000 SU, which has 3 different flow paths, 30 to 300 ml per minute, 1500 ml per minute, and 3 liters a minute, which can take you to about 30 centimeter diameter columns. The 10,000 SU, which is a 10 liter a minute system, which will take you to about 60 centimeter diameter columns. The 20,000 SU or 30,000 SU system will take you clear to 1 meter columns. And then this is now conventional columns. We can certainly use these with radial flow, Sepragen's radial flow columns as well. But the systems are column agnostic. They can run with anyone's columns. And then we also have 60 liter per minute customized single-use system, which will take you to 1.4 meter columns, diameter conventional columns. Okay? So it's very important that there be scalability. And then the other pitfall, another question to ask is, how easy or difficult is it to change out the flow path, right? Sometimes other folks' flow paths come in multiple parts, and they have to be assembled in a clean room, and that takes time, and then they have to be installed, often taking as much as an hour or more. And with Sepragen systems, the entire assembly is done under 15 minutes. And so there's a short video. This is about 8 times the speed, but it just shows you very quickly that the operator can simply take a pre-assembled flow path, and there they're putting in tubing that goes into the pinch valves, like that, and threading it through, the flow cells through static sensors, and they can very, very quickly assemble these things, and we've timed it so it's done under 15 minutes, right? And this is just an amateur video. It's not, it's not a professional video that's been taken. It's very simple. We use it in-house for training, and certainly use it for customers training as well. It's not like this is 8 times faster. We've accelerated the video just for the purpose of this presentation, but it just gives you an idea of how quickly you can actually install and how easily the flow path is actually installed. So I'll stop here with that comment. And should somebody be interested, we can certainly meet with you separately and go through these in more detail, right? And of course, the 7th pitfall is, can your single-use system do inline? Can they use buffer concentrates? Right? So instead of making buffers each time, you could make a stock concentrates. You could use, 10x concentrate for your equilibration buffer or wash buffer or elution buffer, and then use the system's blending capability to dilute these with Wi-Fi and get 1x buffer. So we can do these. So our systems enable you to do this, and you can do inline dilutions, and you can target a conductivity value. So you could start with like 200 millisiemens, for instance, salt solution. You're doing an equilibration buffer here at 10 millisiemens, or a wash buffer at 40 millisiemens. And the beauty of this is it's the same program, just going from this bench scale system to as large as a 60-liter system. And we get the same kind of performance on small scale as well as large scale, simply by changing the flow rate on that particular program, right? So, so this capability, these questions are very important when one is looking at going to single-use systems. What is it that you're actually going to be looking for? What capabilities can your system provide? And so, for instance, with our 3000 SU system, we have 3 interchangeable flow paths with different flow rate ranges. So 3.42 millimeter, 6.4 millimeter, and 9.6 millimeter, going all the way from up to 300 mils a minute, 1500 mils a minute, 3 liters a minute. And all of these come pre-sterilized and can be changed out in about 15 minutes. This is an inside look at one of our 300 SU, the small system, which is the 0 to 300 mils a minute system. And you can see the layout of the various single-use sensors and flow meters, etc. And then we have another video showing the 3000 SU system. Again, I won't play the whole video. These are available. Should you be interested, we can send you some of these videos, but it'll just give you an idea of the capability. And some of these are available on our channel.
Welcome to this introductory video about the unique Quanticept single-use system. The SU features all the benefits of the traditional Quanticept system, including some innovative design features that are optimized for even the most demanding production environments. The SU's core fluidics is a completely interchangeable sterile fluidic pathway. The flow path contains all the sensors required for a complete analysis and processing of your sample. The single-use sterile pathway completely eliminates the need for complex cleaning validation and can be exchanged in approximately 15 minutes. The flow path has a complete sensor suite that includes pressure, pH, temperature, and conductivity sensors, and features a twin wavelength UV cell that covers the 254 and 280 nanometer range. Being single-use, the replaceable pathway completely eliminates the possibility of carryover or contamination from run to run. The fluidic pathway is controlled by a series of pinch valves which completely eliminate the need for membrane valves or fluid manifolds that can be a common source of contamination. Gradient control is accomplished by 2 Watson-Marlow peristaltic pumps via a static mixer, 2 flow sensors and an inline pressure sensor. The industry standard 316L stainless steel housing and its components are completely shielded against water with an IP67 water resistance rating. In the unlikely event of a fluid leak, the base plate has been shaped so all the fluids are directed to a central leak sensor that has a threshold trigger of 3 milliliters or more, automatically shutting off the run and alerting the user. Though the hardware is significantly different from the standard Quanticept system, the software layout and logic remains identical, maintaining the same intuitive, high quality and highly functional software that covers the Quanticept range. The software displays intuitive but complex information and guides a user through the method development steps, which is underpinned by an FDA 21 CFR Part 11 compliant user management database that keeps the data secure and fully GMP. With its intuitive software, automated runs are extremely simple to develop, load, and run using a clear and effective representation of the workflow to help guide the end user. Your imagination is the limit, not the program. The main screen of the software displays the flow path as an interactive graphic with all the valves, detectors, and sensors displaying their process values. The pumps can be set to the desired flow rate and will activate as soon as the full pathway is opened. When a manual run is started, all parameters and process values will be recorded to the database. Generating reports has never been easier. Using the process values and data sets from the run, reports can be generated with just a few clicks and is completely customizable to the exact user requirements. Peak analysis is via the software's integrated peak analysis tool. This reads out and represents all relevant peak data such as height, width, integral, theoretical plate count, and height equivalent theoretical plate count.
So I'm going to stop here and not continue with the rest of the video. You get the idea that this is very intuitive, very simple, and highly capable software. And it's the same omnibus software that's used for our single-use systems or multi-use systems, buffer blending, buffer dilution system, systems integrate, continuous systems, systems integrated with buffer blending, etc. Okay. And so we just learned about the 3000 SU and the different flow paths that they have. And then these are, again, we have single-use systems in continuous multi-column, right? SMB or simulated moving bed configurations as well for people who are interested in, getting more productivity out of their process. And then our software is sophisticated, so we can communicate with anyone's DCS systems, with Profinet or OPC UA. We have login authentication, CSV. So if somebody wants to, connect through their Windows permissions into our software, they can do that as well, right? And many alarms settings, customized settings that we can have for not only pressure and air leaks, things like that, but for process values. So you want conductivity within a certain range or pH within a certain range. Outside of those ranges, the buffer can be ejected, or you can get a process tolerance alarm. Those kinds of advances have been built into these single-use systems. Just give you a few screenshots of these systems. Methods are very, very easy and intuitive to write. They're designed around the look and feel of an Excel spreadsheet. So each row in an Excel spreadsheet, if you can imagine, is a chromatography step. And in the table, you simply fill out, in each of the columns, you fill out the valve number, the flow rate, the change values for the step or the change criteria, whether it's time, UV, pH, column volumes, or even Boolean functions, so time and UV, like that, or mimicking like a watch function, right? Certainly ability to do gradients. So all the common things that you're used to with other systems and more can be done with these machines. Data analysis, peak analysis, gradients, blending, and you can cut copy steps. So this is a simple program, like a 7-step program. They are changing based on bed volume, say 1 bed volume or volume. You can have UV, pH, conductivity. Here's the direction of flow in the forward direction. So you're loading from valve pump A1, and you can have your list of buffers and click on them. Flow is 49 mils per minute. And then at the end of the step, you can pause. You can, go, if you have a pre-column filter, you can tell it to go through the filter or bypass the filter. If you have an injection loop, you can tell it to go through the injection loop. So lots of capability. We can discuss in more detail should any of you be interested. And all kinds of reports, right? So method report, user report, we have different levels of users that can log on to the machine. Some people have permissions only to run methods. Other people have permissions to make methods, or some people can only, metrology folks, for instance, can only do calibrations, which are done from the system, the high, low points of all the sensors, or, event reports, and journaling, right? It's very important that if there's data that's deleted, that track all that for 21 CFR Part 11 compliance. Gradients, again, we have up 5 to 95% gradients. Other folks with the kind of low end that they have on the pumps can't give you that level of linear gradients. And then there are, of course, I'm not going to go into details of how our machines compare with other machines out there, but there are definite advantages in terms of performance and holdup volumes and accuracies, etc. And then we can customize systems as well. So we do our complete own skid design here at Sepragen. We have the world-class engineers, electrical, mechanicals, our own software development, our own manufacturing, our own 3D printing, our own electronics area and clean rooms, machine shops. So certainly look us up. We are happy to work with you and customize to your needs. And we do everything from a customized URS to design work, to project management, IQOQ, SAT, FAT, training, etc. And of course, ongoing PMs. So why would you work with us? Of course, we have superior technology. We've been in this field for 30 years, and we understand a lot of the issues that people are facing as they scale up from the bench to large scale. And we also understand that what may be applicable for, let's say, a bacterial product purification chromatography system may not be suitable for mammalian or may not be suitable for viral vectors, and certainly not for mRNA. We've developed what's not shown here, maybe the basis of a future topic of conversation and presentation, is our Camerna system, which is a continuous mRNA manufacturing systems, which goes all the way from IVT to final product, affinity capture, and then separation of the single and double-stranded mRNA. And our systems are being used in gene therapy for people doing, viral vectors, whether it's from AAV, whether it's the ion exchange step, or, some multi-mode step, to folks doing lentivirus, right? So all kinds of applications. And with that, I will conclude right now and open myself for any questions that anyone may have. So any questions? I see there's some questions. There's something in the chat. Sanjit, could you tell me what that is? Any questions in the chat or just remarks?
No questions in the chat.
No questions. Okay. Well, you know what? Sometimes people are uncomfortable asking questions in an open sort of forum like this. I would request that you please reach out to us at our email address, or through just our contact box on our website, which is www.sepragen.com, and schedule time for a one-on-one, and we are happy to do a special Teams call with your team or you individually and discuss your application and your concerns and your challenges. Would welcome that and be certainly very happy to work with you. So again, thank you for joining us this morning. And I know it took something for you to be here, and we appreciate your interest, and we appreciate your being part of our extended network of people that we work with. I think you'll see really exciting, amazing things coming out of Sepragen. We believe ourselves to be technology leaders and really are excited to work with you on your particular application. So thank you again, and goodbye.